This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 32 for May 15th, 2009. From New York City, I'm Vincent Racaniello.
And from Western Massachusetts, I'm Alan Dove.
Joining us today is Raul Rabadan, who is an assistant professor of biomedical informatics here at Columbia. Welcome to TWiV, Raul.
Hey, it's a pleasure here.
Raul, I discovered only last week. He's been here at Columbia since September, right?
September.
And he published a short paper on analysis of the influenza virus, the new H1H1 sequences. So I thought we could have him on TWiV. He was gracious enough to join us, and so we really appreciate that. So you are doing bioinformatics, but in fact, you're a theoretical physicist. So how did that happen?
Well, it's kind of an interesting story. It was I did my PhD in theoretical physics, and I was working in Geneva at CERN, this a collider of particles there, and then for a couple of years, and then I moved to Princeton, to the Institute for Advanced Study. And I was working in physics, and then I was talking to a friend of mine, another physicist, that started to work in HIV, about some interesting statistical problem about the sequence of HIV. And then I started collaborating with him, and I like it a lot. And at some point in my career, I was deciding, well, I think this is very interesting. So I decided just to follow this path, and this is what I'm doing now.
You got a PhD in Spain?
Yeah,
in theoretical physics. And
theoretical
then
physics.
Came to the institute and then got seduced by viral bioinformatics.
Yeah, I was 3 years working in physics and another couple of years working in biomedical,
so
bioinformatics.
There's no more going back to physics?
Not much. I talk to my friends in physics. I love talking to them, and I love physics. But
so if you hadn't gone into virology, what would you do in theoretical physics?
What I was doing before, it was particle physics and string theory. This is basically what I was doing in my PhD and afterwards. Yeah.
So you would probably have an academic job doing research.
There was a point in my career that I had to decide going back to Europe or staying here and going back to Europe working in physics, or staying here working in biology.
So how did you end up at Columbia?
Well, I like New York and I like Columbia.
Okay. You were looking for a job, I presume. You didn't want to stay at the institute anymore, right?
Yeah,
Well, this is a good example of how the many different ways you can get into virology. As we know here on TWiV, virology is fascinating, so we attract a lot of people from different fields. But, informatics is very important now for virology because we are generating sequences at an unprecedented rate.
Yeah, and it's increasing now with the new technologies.
Yeah, and I think in this outbreak, this influenza outbreak, the amount of sequence is amazing that's being generated.
Yeah, today there were more than 120 sequences already deposited in CBI.
So we need help in analyzing them because my skills are very poor. So I'm glad I know you're here because when I have a tough sequence problem now, I know where to go. Well, we're going to talk quite a bit about what you do with sequences. But before we do that, I just had 3 stories that I wanted to bring up, and then we'll move into influenza. And these are virology stories of the week. And the first one is interesting. It is about a woman, her name is Martha Mason, who died last week at 71. And she had spent 60 years in an iron lung. And she went into this iron lung when she was 11 years old because she got polio. And it paralyzed her breathing apparatus, and they put her in this iron lung. Now, back in the day when polio was quite prevalent, a fraction of the people would have their lung muscles paralyzed because the virus would go into the brainstem and it would destroy the breathing center, basically. And so there were many, many people who went into iron lungs, many of them transiently, but some never came out. They were never able to gain the function of those muscles again. And this is one example. So she was in for 60 years. She lived her whole life basically in an iron lung. Oh. And it says in this article that there are probably 30 other people in the U.S. who are still in iron lungs full time. And I think it's amazing. I think at some point we have to step back and realize that viruses, we look at viruses as amazing things, but they do things to people that aren't very nice. And this is one of them. Of course, they kill people, but being in an iron lung And in this article, which we will link to, which was in the Times this week.
It's an incredibly poignant article.
Wow, it is. She said she felt on September 1948, she was 11, she went to bed feeling achy, but she didn't want to tell her parents because they had just buried her 13-year-old brother who had died of polio a few days earlier.
What a story.
So it's very poignant, yes. I mean, this is beyond the science, but it is one of the viruses that we talk about. So we'll put a link to that. And it is very interesting. There's still people in iron lungs, which I guess is amazing to me. I thought there would be ways to get around that, but I suppose
I think there are, but it's a question of what the patient wants. I gather in this case, she didn't want to be on a respirator that would require more sophisticated nursing support. She liked the fact that the iron lung was low maintenance. So she stayed on it.
It's interesting. I guess the 30 others have similar choices as well.
Right.
Anyway, take a look at that. It's one effect of a virus that can last your lifetime. The second story I wanted to mention, last week we talked about bocaviruses. You must know bocaviruses, Raul. Have you heard of those? No? These are new parvoviruses that cause human respiratory and gastrointestinal infections. And we talked about an article last week. So this is all done by DNA analysis. So you get respiratory secretions or stool, and you find new viruses. So we talked about a paper last week where an Australian group had found in, I think, children with gastroenteritis, they found this new bocavirus. So I got an email this week from Amit Kapoor, who reminded me that he has also published a paper actually last year in December of 2008 called A Newly Identified Bocavirus Species in Human Stools. So this was also the same kind of analysis, viral metagenomic analysis to identify this new parvovirus in human stools. And I mention this because Amit is also the discoverer of cosavirus, which is another new picornavirus in stools of Asian children. And he discovered that as well. He's out in California, and he's discovering viruses left and right.
Now, bocavirus is not named that because it's an oral virus, is it?
This is a good question. Do you know why it's named Boca?
I don't.
Because it was originally a respiratory virus. Let's do a quick Google.
According to Wikipedia, it's derived from bovine and canine.
Oh.
Okay, good.
Two known hosts for other members of that genus.
So that's not mouth, which Dick told us last week. And Dick is not here, so we can't correct him.
Right.
Dick Despommier. Anyway, a newly identified bocavirus in stool. Journal of Infectious Diseases. We'll put a link in the show notes for that. And then the third virus story of the week is amazing. This has to do with mimivirus.
This is really cool.
And we've talked about mimiviruses. You know about mimis?
Yeah, I know.
This is a bioinformatic headache, right? Yeah. 1.2 million base pairs of DNA. This is the biggest known virus. I guess we have to say known because there could be bigger. But anyway, this is the...
Hey, we could be inside one.
Could be inside a virus. That's true. Well, the whole galaxy is actually just a marble when somebody's playing with it, you know.
That's right. Yeah.
The biggest known virus. I just read something somewhere. Someone said that this is like way overkill for a virus. Who are we to say? But the paper that came out this week is the cryo-EM structure of mimivirus. And this is a capsid that is an order of magnitude bigger than any known capsid so far.
How big is it?
It is 1,000. All right, so this is the problem. The capsid is 5,000 angstroms, and then it has these fibers that stick out, which are 1,250 angstroms, for a total of 7,500 angstroms. And so that was one of the problems in doing structure that has these fibers that interfere with cryo-EM. So in this paper, they found a way to take them off without destroying the capsid. And they do cryo-EM, and the resolution is...
better than 65 angstroms?
Anyway, so now you can see the architecture of this virus. And they have some very nice pictures in this paper. But the neat thing is that there are 20 icosahedral faces, which you would expect. They have hexagonal arrays of depressions, as shown by the cryo-EM. Each depression is surrounded by 6 trimeric capsomers, and these are similar to other icosahedral capsomers. The difference here is that whereas in most viruses these are hexagonally close-packed, In mimivirus, there are vacancies at the depressions with neighboring capsimers differing in orientation by 60 degrees. So there's some slight differences in the orientation of these capsimers, these hexamers. But anyway, it's a beautiful reconstruction. They have very nice pictures in this paper, which we'll link to in the show notes. I think that's a very important finding here. This is from Michael Rossman's lab at Purdue.
Yeah, and apparently the asymmetry of the capsid may one of the things they hypothesize pretty reasonably is that the asymmetry of it may help align the genome within the capsid in a particular orientation.
Mm-hmm. That's right. And that's always right, and that helps deliver it too as well. The starfish-shaped feature of the organization of the nucleocapsid might regulate delivery of the genome to the host, which is a problem that all viruses put forth, how to get the genome directionally into the cell.
Right.
Which you may remember from your thesis work, Alan.
Yes, I remember something about that.
Yeah, he does remember. We remind him constantly. Okay, so those are our virus stories, and now we're going to talk about flu with Raul. But before we do that, I want to remind you of our friends at Citrix who make gotomypc.com. And I have to tell you, I sit in rush hour traffic every day. This morning I sat like an hour getting past an accident. And I think about all the time and money and energy I'm wasting just to get to my office so that I can work on my computer. Whereas I could do it from home with gotomypc.com. And if you feel the same way, that you have some days you could stay home and work from home, GoToMyPC lets you do that. You can access your office PC from home or anywhere in the world. Even from Madrid, you can access your office PC. So if you go to Madrid, you could access your PC from there. It's a form of remote access. Do you work from home at all?
Sometimes.
But you like to come here?
Night.
At night? Yeah, at night.
But you should not try to access it while driving.
No, don't do that. GoToMyPC is brought to you by Citrix, the award-winning remote access service. It instantly lets you access your programs, files, emails. Basically, you log into your computer and you see your desktop. And you can run all the programs on your desktop. You can access your email, your network, and all you need is an internet connection. It's very easy to get going, no hardware, just a quick automatic setup from their website. And it is very secure. It's as secure as online banking. I use GoToMyPC. I love it. So you can try it free for 30 days, but you must visit gotomypc.com/podcast. That's gotomypc.com/podcast for a free 30-day trial. We thank them for their support. There was an interesting article in Science, Raul, which you probably saw this week about this, the pandemic potential. And I want to talk a little bit about that. But let's go over the numbers first. As of May 15th, the CDC says in the U.S., 47 states now have confirmed cases, 4,714 cases and 4 deaths. And internationally, let's see, WHO as of today, 34 countries, 7,520 cases, and 60 deaths. That's Mexico mainly. Mexico has 2,446 cases confirmed and 60 deaths. And the map that's at WHO is very interesting. It shows most of the activity still in the northern hemisphere. We have some cases in Brazil, Argentina, Australia, and New Zealand.
We have to take into account also the sampling here.
Yeah.
Because in many places probably they are not sampling. So, for example, if you just look at how many sequences do we have from Africa, for example, in the last 100 years, there is nothing.
Is that right? There's zero influenza?
There are basically nothing. I mean,
is there influenza in Africa?
I guess it is. I mean, that we don't sample it doesn't mean that it's not there.
Why don't we sample it, do you know?
No, because probably nobody's.
But how about South America? Do we have good sampling there?
There are some sequences, but not many. Not many.
Well, that's a problem with flu, is that unless you have laboratory confirmation, it's very hard to tell flu because it's a very generic kind of diagnosis. Fever, cough, sore throat, And in fact, flu-like symptoms, that's right. And in fact, many of the cases don't get confirmed. And in the Mexican outbreak even, they're not all going to be laboratory confirmed. So you end up having just a number that you're never sure about. I think the idea is that in the next months now in the Southern Hemisphere, it should take off. So we'll see. This science paper was quite interesting because they estimated 23,000 infections in Mexico by late April. So that's their estimate. And they think basically the outbreak in Mexico is over, I think. If you look at the graph at CDC, it's gone up and it's come down again. And
there is something about the 23,000 is that there's many assumptions in the calculation of these things, and the error bars are big. I mean, if you see these, I think it's going from a few thousand to 40,000 or something. So there are many assumptions in these calculations, and I think this is a very nice paper. Results probably they're preliminary, so has to be careful.
Yeah, I mean they do say that this is very preliminary, and there are a lot of assumptions, and you're right.
But it's an excellent paper. I like it a lot.
So they have 0.4% fatality rate based on these numbers, which is not so high.
They say that it's comparable to 1957, to the Asian flu. That was pretty bad.
Yeah, I mean, 0.4% is a lot of people when you have millions of infections, obviously. You don't want to do that. And that's the goal of all this activity, is to try and prevent fatalities. One of the interesting things I thought they did in this paper was to try and estimate the start date of the outbreak.
Yeah, that's very interesting, yeah.
So they had the first cases in La Gloria, which is in Veracruz, I think, right?
I think so.
And that was February 15th, 2009. And then they make this statement: an alternative approach to estimating the start date is to look at the diversity in the genetic sequences of samples collected from confirmed cases, assuming that diversity accumulates according to a molecular clock. So what does that mean?
So, there are several web pages where people have been analyzing these things. So, flu is a virus that is, like many RNA viruses, is replicating and is introducing mutations. And these mutations accumulate, and very fast. So once you sample a few viruses, you realize that they are not equal. If you know how these viruses are evolving, so this is the molecular clock, how many mutations do you have per unit time, then you can infer basically what is the time to the most common ancestor. And from there, you can estimate basically what was the first virus common to all these viruses that has been sequenced in the last few months. So yeah, they are giving some numbers that is in the beginning of 2009. And I think this is a very interesting method. I mean, they have their web pages where analyzing these things. Some of the authors of this paper that they're analyzing these things, they have very nice analysis, and other people analyzing, trying to extract when was the starting day. But there are many, the error bars they're very big. Yeah. So we don't know exactly. You can see that some people they are saying that it's from July 2008 till basically now. So there are many assumptions in these models, but I think they're great because they give you some intuition of what's going on. For sure, it's not a virus that has been there going around for years in humans and been sequenced or something. Probably it's very recent, so perhaps a few months and maximum a year.
Yeah, it's interesting to think that it may have been there in January or even at the end of last year. We didn't know.
Yeah,
it's causing flu, and it's just not diagnosed. They don't look for the strains, I guess, and they didn't pick it up.
Yeah.
So when you do this clock analysis, what do you compare the sequence? So you can know the rate of evolution if you look at all the isolates in an outbreak. But how do you figure back to where you think it started? What is that based on?
You have to basically assume something that is called this molecular clock. So how many mutations do you have per unit time? There are several ways of estimating these things. You can see that basically in different flus, they have similar numbers. And given that you accumulate basically one mutation every replication or something like that, then basically you can go back. You have to see how these substitutions they're appearing. And then you can infer basically looking at the diversity, how many different viruses do you have and how far they are from each other. Can just go back and try to infer that.
Now, do we have good evidence that the molecular clock would keep ticking at the same speed even when the virus is in the process of making a species jump like this?
I think this is an excellent question, and we don't know. One has to do some assumptions, and what they're saying here in the paper, they're saying, accumulates according to a molecular clock, is assuming. So It's basically an assumption. So it's true, what you were saying. So if this virus has been in swine, for example, before, and jump into humans, probably there is a lot of positive selection, and not only at the level of amino acids, but it can be RNA structure, or it can be another things that are changing. So we know that there are a lot of pressures there that are not only amino acids. So this is why these numbers, should be taken with a little bit of carefully.
Right.
So, but they're very good first approximation, and I think they're great.
Oh, sure.
They're great.
Yeah, I think the amount of information we're getting about this virus in such a short span of time is just amazing. It's really, really great.
Yeah, I think everyone is doing an excellent job, and you see the sequences, and they're coming from everywhere, and they're great. They're great.
So what good does it do us if we know it began say in January? How does that help us?
So, from practical point of view, I think there are several things that can be interesting. Imagine that you have the opposite. Imagine that you can see that the common ancestor happened 2 years ago. So then you know that probably it's not coming from this particular place, that probably has been everywhere in the world before.
Okay.
So that's interesting. So if in the other direction, I think it's kind of very interesting from a public health point of view.
So does the fact that let's say it started in January, does that mean it was likely originating in Mexico?
I cannot say. I mean, it just
could be anywhere else.
It can be anywhere else.
Well, I mean, doesn't the large amount of cases in Mexico imply that was the first?
I think probably it's likely that it's coming from there, but I think it's too early to say because, I mean, we don't know many things about the origin of this guy. So,
sure.
I mean, people are looking at swine and different pigs, and they haven't found any yet. So we don't know where it is.
Also, as you were just mentioning, there are many other areas of the world where surveillance is essentially nil, so it could have...
That's right. That's right. I mean, there are continents that there is no sequence from any swine flu. So, I mean, we don't know what is going on. And I think there are two keys for this reasoning to work. The first is that the surveillance is not perfect. I mean, it's not uniform. And the second one is that swine viruses, they're not like human viruses. That human viruses, you go to New Zealand and they're very similar to the ones in New York because they move very much. But swine viruses, they're not like that, like avian viruses. They are more concentrated in some locations. So we don't know. I think it's early, and I think the solution to this enigma is coming from more surveillance and getting more samples from everywhere.
Yeah, not just where we're getting them. We need to expand it, right?
Yeah.
It would help. Now, in the past just a few days, there was an article in Bloomberg, and the title here is Swine Flu May Be Human Error. WHO investigates claim. And this is from a fellow, Adrian Gibbs, 75, who is a scientist. I mean, I looked at his PubMed, and he's published a bit on viruses.
In plants.
Plant viruses.
Well, he has a paper in Nature, I think, at some point about the 1918, isn't it?
Yes, he does. He has something about, yes, here it is. Molecular virology was the 1918 pandemic caused by a bird flu. Yeah. And he basically says that there's evidence that it had a mammalian.
Yeah, it's not very clear, and I think it's a very interesting question. I mean, it's not very clear what is the origin of this virus, and there are several hypotheses.
So he is a scientist who he's published some papers, and now he is saying he thinks this is the simplest explanation is that it's a laboratory escape. And then he says, but there are lots of other explanations.
That doesn't seem like a simple explanation at all.
Yeah.
Have you seen any of these data, Raul?
No, I haven't seen the paper, and I don't know what is the evidence for that.
Have you seen anything further, Alan?
Just bits and pieces of the story that are floating around. I mean, apparently he's drawing this conclusion based on he analyzed the sequences and decided that it had more lysine codons than usual for a flu virus, and so he decided it must have been grown in eggs, which that sounds like an interesting hypothesis, but it's an odd way to go about analyzing it.
Well, I mean, one of the things that is interesting is that influenza viruses replicating in different hosts, they have different mutational bias.
Right.
So, and I mean, one can try to extract some, but the statistical errors are big, and it's very hard to estimate how long and numbers and all these things. So, but I haven't seen the paper, so I cannot comment on that.
Yeah, so it says here, CDC has looked at the data and decided there's no evidence to support his conclusion. So we can't even decide here on TWiV because we don't have the data.
Right.
But I have a problem because he shouldn't be going to the press with this. He should publish it.
Exactly.
Have it accepted, because what journal will take it now, because the embargo is already broken, right, Alan?
Right. Yeah, nobody wants to be publishing this 2 months from now.
Yeah. So I think this is irresponsible science, if that's what he's doing. If you call science what he's doing, it's irresponsible, because he should have it peer-reviewed first, and then now he may think that this is really important.
Yeah, it's a big claim. I mean, it's
so.
Well, but if you think something's really important, I mean, you push it out to some place like PLOS One or one of these journals that has a fast track and a fairly straightforward process, and is going to put it online. Or heck, put your raw data on your website. And just
no, that's a good point. You can get things out in a week, basically, now, right?
Mm-hmm.
There are a lot of fast trackers. If the journal is interested in it, they'll do it. So I don't know what to make of this, but maybe when we see the data, maybe if it comes out in a couple of months, you can come back and look at it with us. But looks like if the CDC says there's nothing to it, then I tend to believe them. And although we don't blindly believe everyone.
No, but extraordinary claims require extraordinary data, and
Yeah.
at least in our initial searches on this, we're not finding any. So
Now, you have looked at these sequences, Raul, right? I mean, is there anything unusual that would say to
No.
you?
They look like viruses that we haven't seen for a few years.
Yeah.
I think what's interesting is that some of the segments, they are kind of far away from the things that we've been sampling in the last few years, but I think probably surveillance. And so there is nothing. I mean, this virus is evolving at a normal rate, and I think it's kind of early to say that it's evolving faster or something. But I don't think there's anything very extraordinary about this thing, so I couldn't find it.
So you published a paper just a week ago in Eurosurveillance about the origin of these segments, which maybe we could discuss. But before we do that, just tell us, what kind of methodologies do you use when you analyze sequences?
So, well, I mean, we use many different things. It depends on what is the question that we want to answer. So there are standard techniques. I mean, there are many phylogenetic techniques, which you just look at similar things. There are clustering. We were using different clustering techniques also for trying to see what are the closest relatives and all these things. Or you can use what everyone is using, the first thing. I mean, just blast, and then trying to see what are the closest using the score of blast. And these things, basically, they give you an idea of what you already know. You have databases. People have been collecting viruses for 100 years, since 1918. I think the first one is from 1902 that is there. And so these viruses, they have been deposited in public databases. And what you do is just you have a new virus, and you look at what is the closest relatives looking at these things. And there are several techniques to do that. And
so do you write your own programs to do this?
Yeah, usually we write.
You do your own
code.
things?
Yeah.
Because BLAST is a public access program.
Yeah, but I mean,
there are other things.
You can use whatever technique. I think in this case, basically, it's kind of a robust thing. I mean, you can change a little bit the analysis. But
yeah. So in your lab, you write your own programs basically
we have,
to do this?
I mean, it depends on what is the problem. We just define new methods to find what we want to do. I mean, unless there is something that is very standard. I mean, like looking at these things or something.
Sure. So what did you find out about the origin that you published in the Eurosurveillance?
So it seems that the closest relatives, at least in public databases, there are things that they are swine viruses, but from two origins. One is North American swine, and the other a little bit distant from Eurasian swine viruses. The North American lineage is related to viruses that were isolated in 1998, 99. They were the triple reassortant. So there was a combination of a human, an avian, and a swine virus that reassort again with the swine virus, and we produce an H1N2. So it's a complicated mixture of reassortments. Is a kind of complicated mixture of different things, and reconstructing the history of these things, it can be a little bit cumbersome. But in this case, I think they are the closest relatives, and I think it's an interesting virus in the sense that it's related to several species in different places.
So the hemagglutinin came from a classical North American swine virus, which has been circulating since 1918,
Since 1918,
right?
Probably, yeah.
So how distant is this H1 from the closest swine virus isolate?
Well, I don't know the exact numbers, but you can do the calculation. So from the current swine viruses, this one to the current swine.
Yeah.
So it's, I don't know, 4% difference or something.
Okay.
A few percent, 3%.
The neuraminidase is from Eurasian swine viruses.
And that's more distant.
So what is a Eurasian swine influenza virus?
So there are several variations of that, and you can see there are many variations, but most of them related to H1N1. They were in Europe and Asia in the 80s and 90s.
Oh, so they're in both regions, basically.
But there are different H1N1s than the ones that were here.
So Europe and Asia, they are different H1s or N1s?
The ones in North America, the H1N1s in North America, they are different from the other ones. Yeah.
But the one's called Eurasian. Is that because they circulate in Europe and Asia?
That's right.
And they're pretty much the same virus?
And they're basically related to that.
So my understanding is the neuraminidase and the M gene come from these Eurasian
Exactly. The
viruses.
closest relatives, they are coming from there.
So this is what's unusual in that these two sets of genes from North American and Eurasian pigs are in one virus. So that has not been detected before, correct?
Well, this particular combination have not been seen before.
Okay. So how did these two get together? Do you have any idea?
That's an excellent question. So I don't think props is such a mystery. I mean, it's just, and these viruses, I mean, even if there is kind of a geographical isolation between the different things, they mix, and you find many cases of swine viruses in, I don't know, in some places in Asia that they have a combination with something that looks more North American. So there is, I mean, it's not a complete barrier that is not separating continents. There is something there. What is interesting is that there's not been found closest relative in the last few years. So we don't know what the ancestors of these guys have been doing in the last few years and where they have been. I mean, this is kind of interesting.
So what you're saying is the pigs don't actually have to mix in order for the viruses to reassort.
Usually you need a co-infection of two different viruses.
Right, but does this imply that somehow North American and Eurasian pigs were together at some point?
Well, that's likely possibility, but we don't know.
But it could also be that a bird brought the virus back and forth, or that's
it
unlikely.
Could be. I mean, you can see that interspecies jumps, they are not as frequent. So, but it could be. It can be a human. One of the things that it seems is that this virus, for example, it has been jumping in humans and perhaps going back to pigs, like in the cases in Canada. So perhaps, I mean, the story can be much more complicated. I think the parsimonious explanation is swine viruses, and we don't know.
So maybe a person also brought it back and forth.
It could be. It could be.
Because swine industry, you can imagine there is some movement of people between.
I mean, there was an interesting. I don't know if you saw this paper. There was an interesting paper last week, and they were reporting like I think there were 12 cases or 11 cases of human getting swine viruses in North America.
Right.
Since 2005, I think. So I think these things happen, and they're human getting swine flus, and so we don't know how often or how likely these viruses they are going to spread to the whole population.
Now, you mentioned that there's a 4% difference between the H1 and the closest.
Yeah, I think it's a 2% or 3%. I don't remember.
How about the neuraminidase M2?
Neuraminidase, they're far away. They are like 6% difference or something like that. And it's related to the viruses in the beginning of the 90s and last part of the 80s, beginning of the 90s in Europe and Asia.
I see. So related viruses are not currently circulating, or at least we haven't picked them up by current surveillance.
At least. They are the closest using, some alignment methods. Yeah.
All right. So that's the Eurasian components that we're talking about there.
Yeah.
Well, that's it. So that's going to be something interesting to sort out.
Yeah, it's kind of interesting.
Are you working on that?
We are trying to find an explanation for that, and we have several hypotheses, but probably the most likely explanation is just surveillance, because the surveillance is clearly not run. I mean, it's not uniform. There are many places that are not.
So how do you do this? Do you wait for the sequences to come out daily and then you grab them and start playing with them?
That's right. So the first thing that I do in the morning is just I take my coffee and I download the sequence and I do a tree.
Nice. You do a phylogenetic tree.
Yeah, I do a tree. I mean, just
with some program you've written.
No, this is basically using what is in NCBI. I mean, just to look at it in the morning, and then if there is something interesting, then I just dig it.
So explain to us what a tree does, a phylogenetic tree.
So, a tree is basically trying to give you an idea of what is the reconstruction of the history of the different viruses, how the different viruses they are connected. So if you take basically the last cases, they're a little bit different. They are not exactly equal. They're very similar. What is telling us that the origin is probably of all these viruses is recent, but they're a little bit different. So you can compare and you can say, well, this virus that we found in California is similar to what we found in New York. And the one in New York is similar to the one that we found in the Netherlands. And you can just basically try to follow and trying to see if you can just reproduce some of the story of the history of this virus.
Now, when you trace these back, do you see any difference in the rate of evolution of this virus as pig farming techniques have changed over the decades, or is that something you've looked at?
I haven't looked at that, but
no, I just wondered if having larger pig farms might have an effect on transmission rate or not.
Well, probably. I mean, if you think, I'm not sure how these viruses are in pigs or how the pigs are protected, but in humans, basically, you clear the virus and you need a large human population to keep a virus circulating.
Right.
So I think your comment is interesting, because clearly, if you have a huge population of a host, you can keep some virus going around that you cannot in a very small population. I think probably it applies to every virus. I mean, you should know that. So, you can do some very simple epidemiological models of these SIR, some models, and trying to see what is the minimal population that you need to keep a virus going around. And so I think probably changing in bigger numbers or changing basically the structure, how this virus is transmitted or kept.
I read that from 1918 until maybe the 90s or 70s, the evolution of swine viruses in pigs was quite slow, and then it changed. It became more rapid.
This is at the amino acid level, you're saying?
Yeah.
Yeah.
So do you understand why that might be? Or
no.
Is it something you've noticed, or you haven't looked at that?
I haven't looked at that.
Yeah, that's at least consistent with the idea that that the advent of larger scale hog farming operations may have changed this.
Yeah.
Yeah, that's my idea that as soon as the hog farming increased exponentially, it got reflected in the evolution of the virus. So I don't know if you could pinpoint that, but I have seen that written in several papers. I'm trying to find one here, but I can't find it at the moment. One thing I wanted to ask you, you mentioned that there are a dozen or so viruses that have jumped from swine to people over the past years, and none of them transmitted.
This is the ones that there was a paper that was reporting these cases. Yeah.
Right. So how do those sequences compare to these? Are they classical H1s?
They are related to the triple reassortance virus, so in the 1990, 98, 99, all these viruses. And they are not very closely related to the new ones.
So they don't have Eurasian neuraminidase and M genes?
They don't.
So those could, in part, explain the transmission among humans, right?
I don't know.
It's association. We don't know till the experiment.
Yeah.
It's a very hard experiment to do. It's all going to be correlative because you can't do human transmission studies. But the real question is, why did this particular swine virus now have acquire the ability to transmit?
Yeah.
That's very interesting, because if we can figure that out, we can get an understanding of transmission.
Yeah, I think it's a fascinating question, and there are many things that we don't understand there. We don't know what are the factors. I think one of the key questions here is that what is the correlation between the genotype and the phenotype of a virus? For example, the phenotype is how easy it's going from one person to another person.
Mm-hmm.
And given what is the sequence, that is something that is now the technology is doing it very easy to get. We cannot say from the sequences how easy is this virus is going to jump or how it's going to mutate or something. So I think it's an interesting question that, I don't know, probably can be better understood in the next few years with more data and more experiments.
Let's say you look at the sequences of viruses from Mexico, and then you compare those to sequences from California and Texas and New York, where we think that the virus spread from Mexico. How close are those viruses in terms of nucleotide differences? Do you expect them to be exactly the same, a few bases per segment or more?
Do you see that, some of the difference there are 25 nucleotides or something like that, differences. You see a lot of changes in amino acids. So as to prove positive selection at the amino acid level is still small numbers.
Mm-hmm.
But this virus is changing and you expect to be changing.
So is there any virus from New York to Mexico that's almost the same?
There are some of them that are closely related.
Very close.
Related.
Which you would expect if one was brought by someone.
That's right. Yeah, you see basically that the sampling is kind of very homogeneous. So it's,
sure. No, you have to work with what you have. But and then, do you see in New York, California, geographic locales? Do you see independent evolution?
Yeah, you see clusters. You see basically that some of the sequences from similar locations they look closely related to other locations. But still, there is a lot of movement. And it still is, I think it's very early because the number of differences are small. So
each geographic location has its own pool of sequences, basically.
Some of the sequences that are coming from the same location, they look very similar.
Right. It's more specific. Because you would expect there to be some selection at different geographic locations because the population is different and so forth, right? And I suppose as you get more isolates, you'll be able to look at that even more. Yeah. This is really an unprecedented opportunity, right?
Yeah, you see how this virus is changing all the time.
It's amazing.
I think one of the things that is fascinating about flu is, the amount of data, and also because you see evolution working. You see, for example, since 1918 till now, this virus has changed more than 10% of the genome.
Mm-hmm.
So this is basically like taking all the mammals, starting from one of them in 100 million years ago, and taking a picture of the genome, how it's changing basically every year. So you see evolution working there. It's just you don't have to do some complicated reconstruction of what is the past.
Yeah.
Evolution works.
No, that's a great point, because viruses have such a short generation time.
100 years in a virus, there are millions of years in mammals.
Very important. That's what we try and teach, but real life illustrates it much better than in a classroom. All right, so here's another thing for you to comment on. There's an article in Reuters. A leading virologist in Hong Kong has described the new H1N1 virus as very unstable, meaning it could mix and swap genetic material when exposed to other viruses. I mean, to me, this is just nonsense because there's no reason to believe this is any more unstable than any other influenza, which we know will reassort when it co-infects.
That's right. That's
They're
right.
all unstable, right?
So what?
Yeah.
So this guy is just looking for a little publicity. His name is Guan Yi, a microbiologist with the University of Hong Kong.
Yeah, I mean, sometimes journalists, they interpret the things in different way. So
You think so? I don't know. I just know Alan doesn't do that.
Well, yeah, I try not to, but
Listen, he said the virus has been around only a few months. It is very unstable.
That's, I mean, probably true. That is a few months and it's very unstable, probably like any other
Yeah,
flu virus.
there's no reason to believe it's any more unstable than any others. So that's just very
unstable. Also, I mean, what do you mean by unstable?
Right. So he said he's analyzed the genetic sequences. Let's see, huge information gap due to a lack of regular surveillance on animal disease. Okay.
That's what we're saying.
Each one of the 8 segments has been seen in pigs in the last 10 years, but we don't know when this new virus appeared. I mean, he's just reiterating everything that we've just talked about. And he doesn't give us any specific information to evaluate why he makes this statement. Well, I just wanted to bring it up because everyone is seeing these stories, and we here at TWiV, we like to set things straight, and there's no reason to believe that this fellow knows anything that anyone else does, and this is no more unstable than any other influenza
it's also
virus.
entirely possible. I mean, from the journalism perspective, somebody might have been working up a story on flu. They called around in Hong Kong because obviously that's a place that's very attuned to the issue, and they got a hold of this guy who's a microbiologist and asked him to comment on it, and so he commented. And I don't know, if you look at just his quotes, he's not saying anything that's way out of line. He's not necessarily angling for a particular position here. He's just saying, yeah, it's a virus. It's been around a few months. It's unstable. Its presence is increasing in the human population. From that, you end up with a story like this. So it's not necessarily Guan Yi's fault that the story came out this way.
They could have taken everything out of context. Because they said, well, what's new about flu? He said, well, flu is an unstable virus. It can reassort, and then they take it.
I think also one of the things is that when we talk with each other, I mean, we assume a lot of things that for us, they are normal. I mean, like a virus is always changing. But for someone that doesn't know about these things, I mean, this is kind of surprising. And you say, well, there are like 20 mutations in this virus. I mean, it's not much, but you see that this is basically something that can scare some people.
That's a good point, yeah.
And the general public, the idea of a mutation is kind of radical.
Yeah. And you see that the virus is changing, and then, I mean, especially about people were scared with this virus that was a combination of different things, and then it's changing. It looks like a monster that is scary.
So that's enough for that. I wanted to ask you a couple of other things sort of related, but not directly. And I noticed you published a paper on bias, CpG bias in RNA genomes, and I wanted to ask you about that. With polio, you can make synonymous changes in the capsid, and if you change the CpG frequency too much, and UPA also, the virus dies.
Oh, that's very interesting.
But for listeners, we're talking about two different pairs of nucleotides, either C next to G or U next to A. DNA genomes don't like to have C next to G because one of them gets
methylation
methylated.
and deamination.
Yeah, so it's bad to have CG. So there's a bias in all DNA sequences against that. But in RNA viruses, there's also a bias, which doesn't make sense because it's not methylated. Yeah. So what's going on?
No, this is basically, we were doing some analysis, and you just take a human gene, take your favorite human gene, take P53, and then you just do some statistics, and then trying to see what are the motifs, the set of nucleotides they are underrepresented. And then you find that basically C followed by AG is basically the most significant thing, surrounded by A's and T's. So this is the thing that is very significant. I mean, statistically, it's a mathematical proof. It's there. And then you open a book in biochemistry, and then you see, well, this is due to 2 processes. One is methylation. So there is an enzyme that is adding a methyl group, and there is deamination that cannot be efficiently corrected. So it's losing an amino group. So fine, this is applying very well for organisms that they are methylating the DNA. So, but then you look at flu, you look at HIV, and basically all the viruses that are living in humans. So it's not only applying to flu, and then you find the same thing. So you find that there is basically CpG is very underrepresented, except for rubella.
Really?
Rubella is not. Yeah.
Oh, I didn't know that.
Yeah. So rubella is very interesting. So, and they follow a law. They follow basically a line, and it's like kind of a universal. You take retroviruses, you take other viruses, and they follow the same line. And we have, I mean, this is an observation, and it's a reality. What is the explanation for that? We have several hypotheses. There can be some structural thing at the level of RNA that they don't like to have some particular dinucleotides. So, for example, T followed by NA is kind of funny in DNA. There is another set of things that these viruses that can be recognized by a set of systems. So we know, for example, that CpGs in DNA can be recognized by toll-like receptors. And so if this is true, then basically if these kind of motifs, they can be recognized in some particular context, then the virus that is surviving is basically eliminating the way of being identified. So there is like a positive selection to eliminate these guys. And there is a lot of other set of hypotheses, the amination enzymes, type of apobex, and all these things. So we don't know what is the explanation, and we are working with some people doing experiments trying to find it out. But it's kind of interesting that is kind of a universal law that Cs followed by Gs, they want to be out of these viruses. And then you see how, for example, when there is a flu, since 1918 till now, has been losing CpGs. You can follow the track. You can just basically see a line with time, how it's following. And influenza B, that has been for very long in humans, has very low numbers.
Oh, so they seem to. Is this true of animal viruses as well as human viruses?
We were looking basically at human viruses, and you know, animal viruses, they were kind of complicated because some of them there's some particular vector that was transmitting it. So we didn't know if it was coming from we should consider a mosquito, we should consider this thing. So we took basically for the analysis, we took basically the human viruses. Bacterial viruses, for example, they are also very different. So they don't care about these things. So you can see that there is a correlation with the host. I think it's an interesting hypothesis, but I mean, we don't know yet what is the explanation.
Well, when I first sequenced polio in 1980, I noticed this.
Oh, really?
And we wrote it. There's a CpG bias, and someone said it's meaningless because there's no methylation of RNA, because it was among the first sequences of RNA
I think
viruses.
it's a very interesting observation because it's the explanation that is working there. I mean, it's universal. It's working for every virus in humans.
Yeah. All right. The other thing I wanted to ask you is about this mutational bias in, say, avian versus human influenza, which you've published on. I wonder if you could just explain that a bit.
Yeah. So basically, we were working on a very different problem, and then we were interested in flu, trying to see if we can say something about what is different about avian viruses from human viruses. And then we were looking at some particular oligonucleotides, so combinations of nucleotides all together, and they can separate human from avian viruses. And we realized that there is a big separation between the two of them. But then you just try to see why there is an explanation, and then you take smaller components, and you can see basically that the nucleotide composition is different. So you can see, for example, the number of U's is different in human viruses, human influenza viruses, than avian influenza viruses. And you can see, for example, viruses that were starting in like avian influenza viruses and going into humans, they have this particular trend and they increase this number. So there is a bias. We don't know exactly what is the explanation for that, again, so there is a combination. It can be that it's related to the CpG, because if you eliminate Cs, the compensated of that, well, you can see that. But it's clearly that every time that the virus is mutating, they have a particular direction. So it's a random process, but there's a probability of occurring something more likely than the opposite. So a C going to U is more likely to happen than the U going to a C.
So by that analysis, what does the 1918 virus look like?
So we compare only avian and human in this analysis, and we cannot say anything about swine. And there were several reasons why we didn't do that. And still, I think it's kind of complicated. But it seems that 1918, it looks like an avian virus. So it's so you can just draw the trajectory of these viruses or how many nucleotides are changing. And then you can see that probably was a very recent avian or other reservoir that looks like avian virus going into the human population in the 1910s.
So then if you track the isolates from 1918 towards the present, become more human-like.
That's right. It becomes more human-like.
U composition.
Yeah.
So you haven't looked at the swine viruses at all?
We look at some swine viruses.
Yeah.
We look, and it's complicated.
Okay.
And it's complicated for several reasons. First of all, there is a lot of interhost reassemblement. So many pieces, like the triple reassemblement, the famous triple reassemblement, different pieces, they come from different parts.
Right.
And then you can track each of the parts. And there are many of them. There's a lot of sequence errors that we find. So it means that, for example, you can do the following. You can, for every 2 viruses that you have in your database, you can compare the distance in the genome, how many mutations there are, how many differences there are, and the distance in time. And there is an inequality that you can prove, that basically the distance in the genome, the number of differences, should be bigger than some particular number times the difference in time. And that should be true for any virus that is replicating with some rate. And you find that there are cases that is not true. And these cases, that means that there are sequences that are contaminations, errors, and things like that. And we found many of them in the databases. Many of them, they were in swine. So you can see that some of the swine viruses that were found in 80s or 90s, they're identical to 1930 at the nucleotide level. And that's basically the most likely explanation. It was contamination in the lab. And other cases that you find is like 1977, that we know what happened in there. So 1977 H1N1 was basically identical, very similar to what H1N1 in the 50s. And so some of these cases, they are known. So there are a lot of funny things going with these ones. So we couldn't find any nice explanation for that was convincing.
Yeah, that you published a paper on that, right? The anomaly in the
the
database.
anomalies, because there are many of them. Yeah, and also, for example, there was another phenomenon that is homologous recombination. So it has been kind of a debate. Homologous recombination is real or not, and people has been trying to look in the lab, and basically it's not found. And then, well, you have thousands of viruses, tens of thousands of viruses. Let's look there. Then you have some method to compute these things, and then you find some cases. And the cases that you find, they are kind of suspicious. So what we are saying there, they are kind of suspicious.
I see.
They are cases and they're suspicious.
So there may not be recombination.
So I think
the
process.
And reunion.
Yeah. So when you are sequencing, I mean, if you're not careful, I mean, you can just take 2 contigs, and so making a contig with 2 different reads that are not exactly the same thing. So there can be many things that in the process of assembly can be funny, and then they look like a recombination. So we don't know what is the explanation, and perhaps there is a homologous recombination, but I'm skeptical.
Yeah, we should point out that this would be different from the reassortment, which just means the shuffling of segments in a co-infected cell, whereas this would actually mean one RNA has sequences from 2 different viruses, the traditional breakage and reunion, which
that's right. This is within a chromosome, and the other one is between chromosomes.
Yeah, so this may not happen because it may be an error. So what percentage of the database do you think is wrong?
Oh, I can give a bound.
Yeah.
There are some sequences, and they are coming from some particular places and some particular hosts.
So you have to be careful.
One has to be careful. So we are trying to work with curated. So we have our own curated thing. And
yeah.
I mean, I think it's happening with everything.
You make your own curated database, yeah?
So basically, when we are doing some analysis and then we find something weird, we go through the database and trying to see what is funny with these things. Last
thing I want to ask you is we have talked a lot about people discovering viruses. We talked about the bocavirus. We've talked about virus. How does someone who works in silico discover new viruses?
So I think there are 2 things that are fascinating that are happening now, and we are realizing that they are happening now. One is new sequencing technologies. I mean, sequencing is becoming cheaper, and there is meta-sequencing. So you can just metagenomics. You can just take a sample from something, getting all the RNA or DNA that you have there. There's going to be a lot of things from the host, for example, if you're discovering new viruses. But at the end, you have a lot of sequences from a virus that you don't know that is completely new. So that's great because you don't have, I mean, before most of the methods, if you're using a microarray or some PCR or whatever method is, you have to have some information about the closest relatives to these guys. So this is starting really discovering new things that have low homology. Then the other thing that I think is an interesting comment is that our knowledge of biology is very anthropocentric. Yeah, it's based on humans and our knowledge of humans. You can see what are the genomes that are there. There's the human genome, the dog genome, and they're basically our closest relatives or the animals that are living with us. Then when you see basically the bacterial or viruses, they are basically pathogenic to humans or related to humans. So everything is, our knowledge of biology has been expanding through, since these things. So this is basically like a Copernican shift. It's just basically going and putting, you just look at everything, and there are things that are not human or not related to human and all these things. So I think it's very interesting. We are going to discover, and we are discovering many new things. So the question there is how, from this huge amount of data, how do you find things and how do you classify the things? And I think this is a very fascinating problem.
So that's why bioinformatics is such a big part of science now, because you have to learn how to massage the sequence. So if you take, let's say, a stool sample from a human and you sequence total RNA, and you said most of it is ribosomal RNA.
There would be a lot of ribosomal,
Over 95%?
Depends on how do you treat that.
Yeah. Some people make particles, so that would get rid of a lot. Then you have a lot of sequences left, and you blast them, basically? Is that you just search a database with those?
There are several ways of doing that, but basically you want to get rid of the things that you already know.
No. So, and then you're left with something that you
don't know.
Then
you get something which you don't know, and then you have to figure it out. So I think this is a very... another thing that I think is very interesting is that many people are doing all this sequencing of many different genomes, cancer, and all these things. So it's a great opportunity to find things that just looking aside and trying to find new things.
So you enjoy getting the sequence in the morning and playing with it all day. That's what you like to do.
Yeah, this is it's a lot of fun.
So for people out there who are into computing, physics, mathematics background, this is for you, because there's plenty of opportunity to do this. You come trained with Raul and you can have a career in viral bioinformatics. Okay, that's great. Any other questions, Alan? You still there?
No. This covered a fair amount of territory here.
Pretty cool, yeah.
Yeah.
All right, we're going to read a few emails. We get emails from our listeners, and then we'll wrap it up here. We have an email from Sophie, who writes, "Hey, I really like this show. I started on my BSc in biotechnology, and you guys are really giving me something to look forward to when our courses are dull. I also heard about you through Futures in Biotech. I have a quick question." Sophie is from Denmark. "I live in Denmark, so there's quite a lot of influenza and other throat viruses going around here. Even though we have seasonality, it's not uncommon to get flu or something else in the summer period. Despite this, it's not normal to get vaccinated, only some elderly people, and I have never heard of anybody using Tamiflu, although I'm sure it's used in immune-competent patients. But it sounds like it's quite normal to use in the U.S. Can you explain the difference?" Well, as far as I can tell, every country has its own vaccination policy, and I understand Denmark, the policy is 65 years and over, or if you have a chronic medical condition, you get immunized. And I know that Denmark has stockpiled Tamiflu in case it's needed for swine flu, so it would be used there. And that drug was actually licensed in 2002. So every country is different. I don't know what would distinguish Denmark from the U.S. The U.S. is a big, diverse population, and they don't behave. They don't do things that, in Denmark, I think the government tells you, stay home and don't transmit diseases, and you listen better than we do in the U.S. So I think we have to have a more broad vaccination policy here in the U.S. I don't know I'm not familiar with policy at that level and why countries do different things. What I do know is in terms of immunization programs, Scandinavian countries, for example, have always done well because people listen, they get their immunizations, and they behave in a good public health manner. So that's all I can tell you. Sorry I don't have a better answer, but I'm glad you like TWiV, Sophie. Jim writes, who's a DVM, "I enjoy your podcast a great deal and have listened to it since December 2008. I recently heard you discuss West Nile vaccines for horses. The first vaccine that came out is made by a vaccine company called Fort Doges. It's a simple killed vaccine. It came out within a year or so of the outbreak of West Nile in the U.S. It isn't an elegant vaccine. About 3 or 4 years ago, another company called Merial came out with a version made by canarypox vector. Now, how often should a horse be vaccinated? Some veterinarians suggested once every 6 months. The science really has not been there, so the practice is just to do it every year." So he sent us an abstract on this canarypox West Nile, which is basically a pox virus vector, which is used to carry West Nile genes. And canarypox doesn't cause disease in the horse. How often? Well, if there are no studies, then you just do it every year. That's the safest thing. I didn't find any studies of efficacy and longevity in horses of these vaccines, so that may be that the simplest thing is to tell them to do it every year. That's what we were talking about a couple of
And
weeks
obviously,
ago.
somebody should do the study. Just take some blood stabs from horses and check their titers.
Yeah, see how long it lasts. Because the horses do live a long time. Do you have a horse, Raul?
No, I would love to.
You would. But you have to.
I would love to.
You have to immunize it every year.
For the flu also.
Is there flu in horse? Yes,
Yeah,
there's horse flu, equine flu. All right, Steve writes, thank you for your interesting podcast. One question that I have not heard addressed is this. Which of the flu symptoms are due to primary damage caused by the virus and which are evidence of immune reaction? How does the virus cause each of the flu symptoms? I read recently that congestion associated with flu is due to capillaries expanding to permit passage of the immune response to the site of infection. If this is the case, it would seem that treating the flu symptoms might make matters worse. Is this the case? Well, as in every virus infection, the symptoms are probably a combination of virus killing of cells and the immune response. And, a lot of these nonspecific symptoms, fever, cough, sore throat, these are actually caused by the cytokine response, the immune response. So the studies have been done with many viruses which show if you take away components of the immune response, you can actually make less pathology in a virus infection.
The disadvantage, of course, is that then you make more virus.
Yeah, so you have to balance it. You have to make sure that you don't kill one for the other. So, for example, with West Nile virus infection of mice, it causes a brain infection. If you take away toll-like receptor 3, which is an important sensor for the innate response, you actually get a less severe encephalitis because the TLR3 stimulates a very bad inflammation. So you have to check for each virus. These studies are ongoing with flu. I have seen a study in mice, for example, which shows that if you suppress the cellular immune response, you get a less immunopathology. So every case has to be evaluated. So you can't make a general statement that says that if you treat the symptoms that you're going to suppress the immune response. It really depends. Right now, we don't have any drugs that treat the symptoms of flu. No prescription drugs. So
do you know? I mean, just a general question. There were some reports that they were saying people were dying from cytokine storms and this flu. So do you know what is the treatment for when you see that things are going out of control?
I think that's mainly with the H5 infections.
With
right?
the H5N1, but there were some reports also of the young people in Mexico.
Yeah, I saw that, but I asked Peter Palese, and he said it wasn't confirmed that it was a cytokine storm.
I see.
I don't know what you would do to treat a cytokine storm. I'm not sure you could find out quickly enough.
Yeah.
Right? It's such an early response. There are ways to suppress cytokine responses, but I'm not sure that they are used in people. Do you know anything about that, Alan?
I haven't. There's been some work looking at that. I mean, the danger is, you do these tests in animals and you find, for example, the one you just mentioned, you can decrease pathogenesis by inhibiting toll-like receptors. But, what if you missed your diagnosis and it's actually a different virus?
Yeah,
or,
sure.
What if you get some secondary effect? There's a huge hurdle to doing these sorts of trials in humans because you could kill people very easily by suppressing their immune system.
Mm-hmm.
So I don't know the answer to that's a good question. I'll find out, and we'll let you know next time. I want to read 2 more emails. One is from Marisano. I know this is way back, but I was listening to TWiV number 8, and I have a couple of comments. About 5 minutes and 45 seconds into that episode, you made the comment about HIV. It is without doubt the most prevalent virus infection on the planet. First, wouldn't HSV-1, even only when considered in America, be a far more prevalent viral infection than all the HIV cases worldwide? Wouldn't the same be true of other herpes viruses, such as that causing chickenpox?
Or Epstein-Barr.
Yes. So I shouldn't have made that comment. I was wrong. It's a lot of people are infected, but herpes probably at least half the world or more. Epstein-Barr, chickenpox, yes. You have to be careful when you make such statements, and I'm guilty of them because it always sounds really good. But you could say it's one of the most prevalent infections.
Yes.
And then he says, I take it that above you are really referring to the number of HIV virions versus those of other viruses, but that statement is quite distinct from one about organismal infection or even cellular infection, since viral particles circulating throughout the blood are by definition circulating and thus not currently infecting a cell.
Now he's giving you too much credit.
Yeah, really. I didn't even think about that. Even with these potential revised definitions and whether or not the distinction is relevant to the formation of new cases, I wonder, do you have any data on the number of dendrites typically infected by HSV or zoster? No, I hadn't been so sophisticated in my thought. I was just thinking of the number of people. Maybe, the number of virions. There are a lot of HIV virions, or genomes, we would say. How many HIV genomes? I think there are 10 to the 16th HIV genomes on the planet. And that may be a lot more than herpes simplex viruses. So in terms of the number of neurons or ganglia infected by herpes simplex, I mean, many in a person, but certainly not to the extent of, say, lymphocytes infected with HIV. We're always going to find exceptions, but I appreciate the detail. Second, it would appear that phages found within E. coli would far outstrip human viral infections, particularly when proviruses are considered, such that the incorporated virus isn't likely to immediately lyse the infected cell because of their vast numbers. So maybe bacteria would have us beaten regardless of the definition. That could be. In fact, there are 10 to the 6th viral particles in the ocean per mil. A million. So I think the total number of bacteria, of viruses in the oceans is 10 to the 30, so that probably outstrips everything. At any rate, thanks. I enjoy the show, and I am learning a lot on account of it. Sounds like you know a lot already. It's pretty good. Yeah. And finally, the last email is actually from John, who wrote it to my blog, but it's very interesting, and it's about flu, so I'd like to read it. Vincent, you have written about the likely source of 1918 pandemic as coming from Fort Funston, Kansas, now Fort Riley. I also presume that Haskell County, Kansas probably means nothing to you. But Haskell County is in far, far southwestern Kansas. I grew up in Stanton County, Kansas, 2 counties west of Haskell County on the Colorado border. My grandfather farmed in Stanton County. His father and others farmed in Gray County, bordering Haskell County on the east. The only thing distinguishing Haskell County as well as Stanton and Gray counties are 1, a stark lack of population, and 2, a significant distance from a population center of any size. I mean, these places are really geographically isolated by most measures. The closest city of any size would be 200-mile one way to Wichita. Camp Funston was 275 to 300 miles away, the state capital 350 miles. Each of those counties' population would have been 1,000 to 1,500 in those days and not much different today. Anyway, John Barry, in his book, The Great Influenza, The Epic Story of the Deadliest Plague in History, discusses the work of Dr. Edwin Johnson, editor of the Journal of Infectious Diseases, who studied the 1918 flu soon after it happened and had published Epidemic Influenza in 1927. Barry opines that Haskell County was ground zero. Barry is the first to have suggested this, but I have not seen anyone really provide a counter-hypothesis. You might enjoy the backstory about a real country doctor as described by Barry and the reasons for his conclusion. And he sends us a link which I'll post in the show notes, to a story. And then he concludes, my grandfather was stationed at Camp Funston, as were most other Kansans who trained there on the way to World War I. I recently learned the lumber used to construct my father's pharmacy in Johnson, Kansas, Stanton County, was obtained using recycled lumber purchased from Camp Funston dormitories after the war.
Right, because there weren't any trees nearby.
That's right. Anyway, it is all an interesting story.
Very.
So, that's great. I appreciate you sharing that with us, John, someone who's close to that epicenter, ground zero. All right, it is now time for our science picks of the week. And I'll let Alan start with his, and then I'll do one. Then maybe we'll ask you if you have one. If you don't, it's okay. But Alan, what have you got?
Well, in honor of next week's ASM meeting, which will be in Philadelphia, and of course we'll be there, I thought of the Franklin Institute Science Museum, which for those of you who are going to ASM or who are in Philadelphia, it's just up the street from where the event is being held. The Franklin Institute is I guess you'd call it a typical big city science museum, but it's one of the first of that kind. It was founded in 1932, I think. And it's in a wonderful old building right in the downtown area. And they do a lot of very interesting exhibits. They have a couple of perennial ones. They're very well known for a steam locomotive that they have indoors that actually rolls on its tracks. It's a full size. It's an actual steam locomotive
cool.
that they brought it in and parked it in the basement of the institute as they were building it. And so this thing rolls back and forth on a set of tracks. And they also have a giant model of the human heart, which is quite famous.
Cool.
It's been there for decades, and you can walk through the heart, follow the flow of the blood. And then, of course, they do a lot of much more modern exhibits with all the whiz-bang technology, but a really fun place to spend a day or just a few hours. And that is my science pick of the week. And I've got the link, and we can put it in the show notes.
That's perfect for Philly because we're going to be there.
Yeah.
And I guess since you live there, you must have visited a few times.
I actually volunteered there as an exhibit interpreter for the 3 years I lived in Philly.
Oh, that's great. Great pick. Excellent job. Well, my pick is an iPhone application called PyCubed from Sunset Lake Software. Now, PyCubed, Sunset Lake Software makes molecules, which we reviewed some time ago on TWiV, and that's an application for displaying molecules on your iPhone and iPod Touch. And PyCubed is a visual math application for the iPhone. It lets you perform calculations as you would on a piece of paper. It uses an animated touch-based interface, and you can typeset the expressions and export them for use in other programs. It comes with a library of over 150 equations. So PyCubed, Sunset Lake Software, we'll put a link in the show notes. It's $9.99. And the things you can get for your iPhone are just growing daily. It's amazing. I would check this one out if you're doing science because it's really great for calculations. PyCubed. Okay, Raul, have you got a science pick for us?
No.
No, there's nothing scientific.
Well, I already talked about the paper in Science the other day that I think it was very interesting about the origin. Also, I think this flu viruses, swine flu infecting humans. This other paper was very interesting.
Okay, so we'll put links to those. One is a science paper, and the other is a New England Journal of Medicine paper.
That's right.
Yeah, that's a very nice paper as well. All right, gentlemen, thanks very much. Remember, next week we will do TWiV Live at ASM General Meeting in Philadelphia. That's Tuesday, 2 o'clock p.m. Eastern Time. If you're going to be there, it's Press Room 203A.
Is that where we're actually broadcasting from?
It is, and we will make our way there. If you're not going to be at the meeting, it's going to be live video streamed at ustream.tv, and we'll put a link in the show notes. It'll be at 2 p.m. Eastern Time, USA. That's USA Eastern Time. We have a website for TWiV, of course, where we have all the show notes and other links, twiv.tv, and that's where you can send us your questions, twiv@twiv.tv. And if you want, you can record your questions and send them to us as MP3 files. That would be great. We'll play them on the show. Raul, thanks for joining us.
Well, thank you for inviting me here, and it's nice talking to you.
Nice to meet you now. I know you're here at Columbia. Don't be scarce. I'll come have a coffee with you now and then. Do you have any website where people can go to find out more about you?
Yeah, there is the one in my group.
Right.
I can just give you the link.
Okay, we'll put that in the show notes. Alan is at Alan Dove at Twitter. That's
right.
your Twitter handle.
On Twitter, I'm Alan Dove.
And dovedocs.com.
That's right. DOVDOX.
Thanks for joining us again, Alan.
No problem. Always a good time.
We'll see you next Tuesday in Philadelphia. I am at PROFVRR on Twitter, and TWiV.tv is the website for TWiV. You've been listening to This Week in Virology, and this week was all about influenza in silico. And I think influenza will dominate viruses for some time until it calms down, maybe till the end, maybe a year.
Probably a year.
This Week in Virology, all about viruses that may or may not make you sick. Thanks for joining us. We'll be back next week live on Tuesday. Another TWiV is viral.